The overall goal of these experimental procedures is to observe the different terroir signatures in the berry metabolome and transcriptome of the grapevine cultivar Corvina.
The case study described here involved the collection and analysis of samples at 3 ripening stages from 7 vineyards in 3 macro zones over 3 growing seasons. The techniques described reveal the prevalent ripening and vintage effect on the berry metabolome. Clear, specific chemical signatures representing the 3 macro zones were also identified in the ripe berries.
The terroir signature at the macrozone level was revealed using suitable statistical techniques, such as the PCA, which is the principal component analysis, and the O2-PLS-DA, which is the bidirectional orthogonal projection to latent structures discriminant analysis, to analyze both the transcriptomics and the metabolomics data metrics.
On the other side, the differences among individual vineyards are more subtle and thus require more sophisticated and sensitive statistical approaches.
Begin the experiment by developing an appropriate sampling plan. Ensure that the sampling plan states the sampling places, times, and the precise sampling procedure. After harvesting 30 clusters from different positions along 2 vine rows, select 3 berries randomly from each cluster, avoiding those with visible damage and/or signs of infection. Repeat this process to obtain 3 independent pools for each ripening stage. Deseed the berries and freeze the pericarp.